I think this is a good time to start. First of all, give me just 1 second. Okay, so good morning, China. We're very excited for your participation at our weekly MSQ Healthcare webinar series. My name is Mark Estigarribia, and I'm the Managing Director and Senior Partner at MSQ. Here with me from MSQ is our venture partner, Dr. Sean Chi, which I believe you all know already. Thank you again for joining us this week. It is our great honor to welcome our special guest speaker and company, Mr. Ben R. Taylor, President CFO at Tyme, to share his and his company's views on how Tyme's technology will fight against cancer. Together with Ben is his team from Tyme, namely Dr. Giuseppe Del Piori, Chief Medical Officer, Dr. Jonathan Eckerd, Chief Business Officer, and Michelle Corfin, Chief Operating Officer. A little bit about Tyme. So Tyme is an emerging biotechnology company focused on exploring novel therapeutic approaches designed to target cancer's unique metabolism. Tyme is advancing proprietary cancer metabolism-based therapies for metastatic cancers. Good evening, Ben from US. How are you tonight? Thank you for joining us.
No, thank you very much for having us. It's really a pleasure to be here.
Yes, thank you. We have tonight in New York City and New Jersey, East Coast, and also in China. Very excited to have you tonight. Before we get into a couple of questions and your presentation from you and your team, we'd love to give you the floor a little bit in terms of saying a couple of words before we start.
Sure, that would be great. And look, we were really happy to be having this call and meeting a lot of Chinese investors, because that's always been a part of our strategy, was to expand out into China. We are a small US focused company right now, but we had always been putting in place the pieces, the IP, the different groundwork that we needed on the regulatory side as well, to be able to make it as simple as process as possible to move over to China. And that's really, as we'll get into the story, and Michelle and John and Giuseppe will go into it in more detail, you'll really see why. China is a very close fit for us. We're a small molecule therapeutic that's broadly effective. It has a differentiated mechanism. So the advantage of that is the mechanism should pair well with a lot of existing therapies. And so it makes sense when you're using, whether it's some of the traditional chemotherapies or some more novel agents, to be able to add on additional agents like SM88. Of course, in our trials in the US, we've shown a lot of the monotherapy efficacy as well. And that's really important because of the side effect profile. So many cancer patients can't take the highly toxic chemotherapies. SM88 has shown to be a real option that has a much lower toxicity profile, but still some very encouraging monotherapy efficacy. So really appreciate you hosting us on this call, and look forward to speaking with all of the investors and introducing them to Tyme.
Great. Thank you so much for that, Ben. So as we get going here, we'd love for you to please comment of yourself, your team's experience, your company's experience in China, both academically and in your career, please.
Yeah, so it's been decades in the making, I guess you could say. So all of us have worked in China and with companies and physicians in China. Mine was a little bit more personal experience. I actually studied Chinese history and politics in college. I studied at Beida for a while, lived over in China during the mid-90s. So I had a little bit more background. Then I moved out into investment banking it was about 15 years before I was going to make it back to China. But then I was the co-head of healthcare investment banking for Goldman Sachs over China and Southeast Asia. So spent a lot of time on the ground, working with companies, working with investors, working with venture capitalists, and getting a much better sense of the market. So for me, it's always been a goal to bring Tyme back to China. From all of the things that I'd learned, I just really was encouraged by the atmosphere and the attitudes about healthcare in China.
Right, great. Just a follow-up, and if anyone else wants to chime in, please interrupt me. What are the excitements and opportunities of doing business in China today?
Yeah, so that really goes back to, I think, what was so encouraging about doing business in China when I was there was people were looking for new solutions. And so I think that brings a lot of creativity into the healthcare system as people try and find new and better ways to treat patients, but also you're doing it in a capital and resource constrained environment. And so that's where something like SM88 works well, because from our cost profile, it's a small molecule that can be more easily manufactured. So you're not talking about a highly complex process. The distribution system is relatively simple. And so you're not involving a lot of the infrastructure that you might need with more complex pharmaceutical agents. So both of those things paired very well for what we would like to do in China. And then I think the last part is just when you really look at how pricing, how approvals are going in China, being able to differentiate product profiles has become so important. And that's where we look at some of the combination work that we could do with SM88 and other agents as being a way to really differentiate both SM88 and its therapeutic benefits as well as the existing agents.
Congratulations on a major milestone achievement with the formation of a U.S. strategic collaboration with Eagle Pharmaceuticals, focused on the co-promotion of your lead CMDT candidate, oral SM88, in advanced cancers. Please comment on the highlighted details and strategy of that major milestone, please.
Yeah, sure. So that was a good partnership for us because what it brought in is really a strategic investor who also has operating capabilities. So we were able to keep almost all of the rights to SM88 worldwide. So we have 100% of the rights outside of the US and 85% of the rights inside of the US. And they made a investment at about, it was about a 70% premium to market at the time. So we always love to bring money in that is less dilutive than an equity financing would be. Plus, they bring a lot of existing experience, especially in community hospitals and other areas, where when you're first launching a product, it's always difficult to build up the sales force in those areas. We have great KOL relationships. The leading pancreatic doctors in the US know us very, very well from our trials and from our work with the different advocacy groups. But having a partner who broadly hits the healthcare system is very helpful. So we were very happy to have them come in, both as a partner and for the capital, and ended up being a great time to do it because it was before coronavirus hit here in the US. It certainly would have been more difficult to get that deal done today. And it allows us to basically sit back and run the business in the way that we should be doing it without having to worry about the capital constraints.
Congratulations again.
Thank you.
Another successful strategic collaboration is with Joseph Ahmed Foundation. Congratulations on dosing the first patient in your phase 2 trial. Please comment on those strategic details with that organization, please.
Yeah, so that really checked off what was an important diversification step for us, but also just a great story. And if I go to the side for a second, Joseph Ahmed was actually a SM88 patient. He was, so Ewing sarcoma, it's an ultra rare sarcoma, mostly affects boys in their teens and early 20s, just a terrible disease that has a very high mortality, especially if they progress after first-line therapy. And so Joseph's parents actually came to Tyme when he had already failed 3 lines of therapy, and they were looking for a different alternative. he was treated as a compassionate use patient. And this is after he was told that he didn't have any other therapeutic options. He ended up having a really nice partial response, getting a lot of his strength and energy back. He actually ended up going to his own prom and doing some of those normal kid things, right, that you'd want a kid to do. And so after all of that, his parents started up a foundation. And basically, they raised money for a couple years for Ewing sarcoma treatments, and then approached us and said, we'd like to do a trial with you so that other families have the same opportunity that our family did with Joseph. And so we'd had physicians, we have lots of physicians who approach us on doing trials in different disease states, and we'll talk about some of that later on. But we had one of the world leaders in sarcoma therapy come to us. He'd actually been a part of the last 5 drugs that were approved in sarcoma. And he had wanted to carry on some of the compassionate use work that we had done as well. We teamed it all up and got the trial started. So it's not just a good story, though, because what we have always wanted to do, in our early testing, we showed complete and partial responses in 15 different cancer types. And so when we chose pancreatic to go after first, there's some great mechanistic reasons, regulatory reasons, capital resource reasons that we'll get into later, but it's really just the tip of the spear. And what we wanted to do was move ahead with the regulatory, move ahead with the sites, move ahead with our clinical programs, and then broaden out into a lot more of those indications where we had had complete and partial responses historically, and then do it in later stage registrational trials. So sarcoma was the first one that we announced and that we've got off the ground and started running. We'll talk a little bit more about it later, but if you look at other areas like breast cancer, hematology, prostate cancer, those are all areas where we have a lot of background, we have a lot of physician interest, we have a lot of different options for bringing it forward. And I think they will follow up in ways that sarcoma, or they could follow up in ways that sarcoma has, in really diversifying out that pipeline, because the SM88 mechanism isn't something that could be used for so many different types of cancer.
Amazing story. Thank you so much. You're a great storyteller, Ben. I love it.
Thanks, Mark.
So one more before we get into the presentation. I'm sure there'll be more collaborations on your and Tyme and your team's horizon. Are you looking for strategic partnerships with China pharma biotech? What is your vision for Tyme's vision for China today?
Sure, it's a great question, and we don't have the perfect answer to it. What I'd say is, it's always been a part of our business plan. That's why we made sure our intellectual property position there was strong, and that our regulatory, the way our trials are run, a lot of that information could be used for foreign filings as well, especially for China. And so it's always been something we've had our sights on. You know, just practically speaking, it makes a lot of sense to do a partnership, because building that infrastructure in China is incredibly difficult for a foreign company. And so we think there's a real win-win situation for us bringing in a proprietary drug that could advance quickly and have a good commercial appeal in China, and for another partner that has the resources. That being said, we have been focused on the US, and I think, you know, it's a good question for us, how quickly we'll move forward, and part of that's dependent on the interest that we see in China.
Great, excellent. So I guess, thank you, Ben. So without further ado, I'll let you take over the screen by sharing, and we can start with your presentation. And shortly after that, we can start with the Q&A session. Thank you.
That's great. So Michelle, do you want to take it from here?
Absolutely. Thank you. Excellent. Well, good morning to those of you in China, and thank you to Mark and the MSQ team for hosting us this evening. I'm Michelle Porfin, the Chief Operating Officer for Tyme. Myself and my colleagues, Dr. Jonathan Eckert and Dr. Giuseppe Del Priore, will now go through our corporate presentation. Some of the statements we will make during the presentation may be forward-looking statements. I would refer you to our safe harbor statement in our most recent SEC filing. So Tyme Technologies, Tyme is a clinical stage emerging biotechnology company that's focused on exploring our novel therapeutic approaches, which we'll talk about in more detail. The platform that we are developing is our novel platform referred to as Cancer Metabolism-Based Therapies, or CMBTs. To date, our focus has really been on both those advanced cancers and those difficult to treat cancers. The investment rationale for Tyme really falls into a few key areas, really 5 key areas. The first is our proprietary cancer metabolism-based therapies. This is a unique platform we have with a very strong patent portfolio, which is broadly covering a number of areas with IP extending beyond 2032. The mechanism of action of this platform, which Dr. Jonathan Eckerd will go through in more detail, it was really developed based on the extensively studied work from Dr. Otto Warburg. About 90 years ago, Dr. Warburg published the fact that cancer cells have unique aspects of metabolism unlike our healthy cells. So our CEO and scientific founder, Dr. Steve Hoffman, developed our cancer metabolism-based therapies with the focus of targeting the cancer cell's unique metabolism, but sparing the healthy cells as much as possible. The goal there was to focus on the efficacy on the cancer cell and also sparing the unnecessary off-target toxicity. We've advanced our lead cancer metabolism-based therapy, oral SM88, into pivotal trials. We were very excited to initiate our first pivotal trial. We refer to it as Tyme 88-PANC. This is for third-line pancreatic cancer. We initiated the trial the end of 2019, and we announced the dosing of our first patient earlier this year, and we are excited to continue to move that trial forward. We were also very proud that we were selected to be part of the Pancreatic Cancer Action Network, very prestigious Precision Promise study. We'll talk about both of those studies in a bit more detail. The Tyme 88 study focuses on third line and is pivotal, and the Precision Promise study focuses on studying SM88 in second line. We also, as Ben indicated in our introduction, have a very important phase 2 focused on sarcoma, both Ewing sarcoma and other high-risk sarcoma. And then the fact that we have such encouraging efficacy across a broad range of tumor types, both solid tumors and hematologic malignancies, part of what we'll discuss for this upcoming year is expanding our clinical trials into other areas, including other solid tumors and hematologic malignancies. One of the areas that we're very passionate about is the fact that the markets that we are studying our therapies in represent great unmet medical needs. These are patients with either no or limited options. So we are passionate about not only starting up our trials, but also making sure that we execute on our trials well. And a very, very key point for us when it comes to patient care is the importance of convenience for the patient. So our lead therapy, SM88, is an oral therapy, which is a very important benefit for patients with advanced cancers. So we have a number of very important milestones for 2020. Some we've already achieved and some are to come. We had the excitement of not only initiating our 88-PANC pivotal study, but also dosing the first patient. We partnered with the Pancreatic Cancer Action Network, or PanCAN, to work with the FDA to finalize that protocol for Precision Promise and get that study up and running. And we also presented our preliminary health economics study at ASCO GI in January. It's important for us, we feel, to partner with the academic community, but eventually to partner with payers throughout the world in terms of understanding the health economic impact of our cancer metabolism-based therapies. So we're looking forward to continuing to advance that work. And some of the other key milestones that will be coming this year include advancing our preclinical and mechanism of action data, and also beginning to publish some of our completed phase 2 studies, including the prostate study, which Dr. Del Priore will reference later in the presentation, and also our completed phase 2 for pancreatic cancer. And also we are looking to advance our next cancer metabolism-based therapy, which we refer to as Tyme 18, into the IND program later this year. Our pipeline at Tyme really falls into three key categories. The first category is oral SM88. It is a pipeline within a therapy. There's multiple therapeutic areas that we've demonstrated encouraging efficacy in. These include both pancreatic cancer and sarcoma, as we've spoken about with ongoing studies. Our prostate phase 2 completed. Dr. Del Priority presented those results at ESMO in September of last year. And we're also looking to expand oral SM88 into other therapeutic areas, and those may include solid tumors such as breast and prostate cancer, in addition to potentially hematologic malignancies. The second part of our pipeline are other formulations of SM88. SM88 does cross the blood-brain barrier. We've seen that with brain metastases from metastatic breast cancer patients, either being resolved or reduced. So when you look at a therapy like SM88, the potential for a nasal therapy for patients with either brain cancer or glioma would be encouraging. We saw some encouraging efficacy in a small number of patients with brain cancers or gliomas. So the thought is that the nasal formulation may even help expedite the delivery even more so than an oral formulation. And the IV formulation is also something that could be beneficial for that patient who is either not able to swallow or maybe digestively compromised. And the third key area of our pipeline is a unique cancer metabolism-based therapy. And the next one that would be in development for us would be Tyme 18. This is an intratumoral delivery. So that would be for that patient that the tumor cannot be resected. It's impinging on an organ, a major artery, a nerve, and the patient needs an immediate therapy to reduce that tumor size. So that was where Tyme 18 may come in. We have presented some very encouraging preclinical data last year for Tyme 18, and we're looking to further advance that compound. When you look at our advancements globally in treating cancers, we've certainly globally made progress as an industry, as academia, but there's still so much more to do. These 6 categories depict just the cancers that we have either discussed or will be exploring. These are incredible unmet needs for the patients. These are also very important market opportunities for companies such as Tyme to continue to explore. We've seen encouraging data in our studies across 15 different tumor types. So these are just some examples of the areas that we have either started studies or are considering starting studies in. So at this point in time, we'd like to share a bit more about the mechanism of action for our cancer metabolism-based therapies and some more of the clinical data for SM88. So with that, I will turn the presentation over to Dr. Jonathan Eckerd.
Thanks, Michelle. So yeah, the foundation of SM88 and how it works, or how it came to be, is our founder was focusing on what Michelle mentioned, is that cancer has a unique metabolism. The Warburg effect, many people know, has to do with energy production, but cancer also changes the nutrients that it consumes to support its uncontrolled growth. And some of these features are ubiquitous across different cancers, hence the broad activity that we've seen. The one that our founder focused on was the consumption of amino acids. So cancer cells greatly upregulate the consumption of amino acids, and specific amino acids at that. And this is to help basically build biomass to support their growth. And what is in the literature and has been known is that there are certain amino acids that cancer cells specifically take up that normal cells in their normal metabolism don't require. And these are non-essential amino acids. So non-essential amino acids in a normal healthy cell can be created by intracellular mechanisms or enzymes that help create these non-essential amino acids from essential amino acids. So I'll give you an example. Tyrosine, which is what the foundation of what ours is based off of, is a amino acid that's not consumed by normal cells. Normal cells create its own via phenylalanine. It converts it chemically to tyrosine, and it supports its own, all of its nutrient needs internally. However, cancer cells across all ranges of cancers have to upregulate channels to pump in tyrosine into the cancer cell. And this is something that's been leveraged in the cancer imaging arena for a while now. And radio-labeled cancer amino acids can image these tumors very effectively because of the selective uptake. And that selective uptake goes back to the comments that Ben and Michelle were commenting on regarding the safety that we've seen with SM88. So we have really seen very little off-target toxicity, and it really follows this biology of this unique amino acid metabolism. So SM88 is a chemically modified tyrosine, and it also is a D and L racemic combination. So the chemical modification is important because it helps increase the affinity for the cancer cell to take it in versus even normal tyrosine. And also the D amino acid is important because if you're familiar with the basic biology, is that our cells are only utilizing L amino acids, and D amino acids have no real biological purpose in most cells, and most cells try to prevent against them. So having the D amino acid is important because it gets into the cancer cell and it causes the disruption of the protein production pathway that is essential for these cancer cells to be able to grow and survive. And so that it's a unique kind of 1-2 effect of having this chemical modification and the D aspect of the amino acid to cause this disruption to some of the core processes that are not only keeping the cancer alive, but helping it to grow and progress. When you cause this disruption, it leads to a catastrophic failure of the cancer cell, and ultimately, cell death and oxidative stress, related, outcomes of this disruption. So it's a fairly basic, kind of simple one, but an area that not many people have focused on with regards to how to target cancer. And so SM88 is really paving the path of looking how to re-examine how to attack cancers through their unique metabolic characteristics. So that's the general gist of how the background of SM88 and the mechanism. And what you'll see from the slides that Dr. Del Piore is about to present is how this has shown activity again across such a range of cancers, because this biology is again ubiquitous across a whole range of different cancers. Giuseppe?
Thank you, John. Yep, so this is our first clinical trial, the first in human trial, which was published about a year ago in Investigational New Drug, and in combination with our Expanded Access Program represent a relatively large cohort of patients. As you can see, this was one of 30 patients at a New York Presbyterian affiliate hospital in New York City. And all these patients had metastatic progressive disease for which there was no curative options. Now, inclusion criteria expected 3 or 6 months sort of survival. And in fact, these patients had nearly a 30 months median overall survival. With that, we saw approximately a 13-month progression-free survival while on SM88 or after SM88 as a single agent. In combination or in total, about 33% of patients achieved a RECIST complete or partial response with a median time of response of about 3 months. And a large percentage of those patients achieved a stable disease. And as we'll see later on in our pancreas data, stable disease is durable and meaningfully associated with survival. Next slide. So here's a summary of what I've just said, 15 different tumor types. And as predicted, we didn't see any cross-resistance with prior therapies, and the mechanism of action that John just outlined would also predict a broad therapeutic application. Next slide.
Excellent. So let me provide a bit of an overview of the pancreatic cancer landscape. Now, this is through a US lens, but unfortunately, the prognosis worldwide is just as poor, which is very unfortunate for patients. So in the US alone, there's over 55 patients diagnosed with pancreatic cancer, and we know that the prognosis is poor. You see first-line therapies in the US are predominantly Abraxane, gemcitabine, and or FOLFIRINOX. That the patients, about a third of patients will respond, but these are not durable responses, and the expected survival ranges from 8 to 11 months. In the second line, you see about half of the patients who make it to second line and are still looking for therapy. Limited agents there. There's one FDA-approved therapy specifically for second line, which is Onivyde. the toxicities for both the first and the second line therapies are very, very challenging for the patients. You're looking at over 50% of patients having serious adverse events, many of them requiring hospitalization. And the expected survival for a patient in the second line is only 4 to 6 months. And then when you reach the third line, there's over 10,000 patients in the United States actively seeking therapy. There's no FDA-approved therapies. There's no data supporting any oncology compendia. Generally, these patients are not seeing any responses to their therapies, and the expected survival is only 2 to 3 months. So there is a great unmet need in the areas that Tyme is pursuing for studying oral SM-88. So by way of an introduction into our pancreas program, based on our first in human study in our compassionate use program, Tyme saw some encouraging data in pancreatic cancer, both encouraging efficacy, but also well-tolerated safety. So we started a phase 2 study. The phase 2 study was intended to be 2 parts. The first part to confirm the dose, and then the second part would have been to take that dose forward into a traditional phase 2. What occurred, though, based on the first part, was very encouraging efficacy, which Dr. Del Priore will walk through. So we convened our medical advisory board, and the medical advisory board felt very strongly we needed to meet with the US FDA to discuss transitioning to a pivotal study. So that's exactly what we did. We met with the FDA in 2019. We received very clear guidance on modifying our protocol, amending our protocol to move from a phase 2 to a pivotal study. This pivotal study will now focus on third-line patients, and it will be a 1-to-1 randomization with overall survival as the primary endpoint. And we'll go through the study design in a bit more detail, but the randomization is SM-88 randomized 1-to-1 to physician's choice. Why physician's choice? There's no FDA-approved therapy. There's no standard of care. So hence, both our medical advisory board and the FDA felt strongly that physician's choice would be the most appropriate control arm. So what I think is very important for you to understand is how did we make that decision in conjunction with the medical advisory board and the FDA to move to a pivotal study? So now I'd like to turn it back to Dr. Del Priore to walk through the data that was initially presented at ASCO GI, and then we updated the data and was presented at ESMO GI in 2019. So I'll turn it back to Dr. Del Priore.
Thanks, Michelle. So here's that data that Michelle just referred to. And if you see look on the left there, we had a cohort as defined in the protocol of evaluable patients, and their survival as of our presentation last year was approximately 6.4 months. Now, if you compare that with other trials that have reported survival in this patient population, the expected or previously reported was only 2 to 3 months. So this is certainly interesting and an encouraging result. If you look on the right side, I alluded to this earlier, where our stable disease is associated with a survival advantage. You can see the blue curve has a significant improvement in survival compared to those patients who continue to progress. And now take a moment to remind people, or for those of you who are not familiar with it, the inclusion criteria was extremely generous and permissive. There was no limitation on the number of prior lines. There was no limitation on the CA19-9. There was no limitation on the number or sizes or location of metastases. And early on, we saw that these patients were quite frail, and many of them succumbed to their disease even between the time of consenting and before the start of therapy. So the fact that they achieved stable disease and that was associated with a survival advantage was particularly encouraging, and we think also associated, with our mechanism of action and what we'd seen in our first in-human trial. Now, on top of that, as I said, this is a very frail population after multiple lines of therapy, and we've reported on this at ESMO and ASCO in some other details. And you could see that it was extremely well tolerated, with only 4% of patients having a serious adverse event, which is very essential in this clinical scenario and in many other clinical scenarios. And of course, this is consistent with its favorable safety profile that we've seen in other diseases and in other clinical scenarios. So after having completed that part 1 of the trial, determining the phase 2 dose, or the recommended dose. As Michelle said, meeting with the FDA and our key opinion leaders, our investigators, the decision was clear that we should move on to a pivotal trial. And this is what's ongoing now as part 2. Very simple, classic, randomized to SM88 versus the investigator's choice, with survival being the principal outcome, and secondaries of progression-free survival and quality of life, with additional key exploratory outcomes, including circulating tumor cells, which we've shown very consistent activity on. Now, in addition to our trial, simultaneously, we had access through our trial and through previous clinical activities to the world's leading institutions, or certainly the North America's leading institutions in pancreas cancer. Here's the listing of the current institutions who are participating in this. Precision Promise is a large patient advocacy group, and they have brought together leading investigators and various stakeholders to advance cancer research using this Precision Promise design, which is a first of its kind, a response-adapted randomized trial. And our drug is the first one to be entering this trial. It's going to be a second-line trial in distinction to our pivotal trial in third line. But it was, I think, a wonderful endorsement to see our work being, in essence, supported by this important patient advocacy group and coalition of leading institutions in North America.
And Giuseppe, maybe if I can just add one thing to that on Precision Promise. I mean, that was really, in some ways, a seminal moment for the company, because we were doing part 1 of our 88-panc trial, so our phase 2 in pancreatic cancer. And one of the KOLs from this group of universities was on that trial and came to us and said, "I love what I'm seeing with the trial. I'd really like you to introduce, or I'd really like to introduce you to this consortium of the top pancreatic sites in the country, because we think that this could be the right drug to launch an entirely new type of trial on." And so Pancreatic Cancer Action Network, for those of you who aren't familiar, it's the largest pancreatic cancer advocacy group in the world. They have actually come in and they pay for a large portion of that first stage of the pivotal trial. So for us, not only was it this great exposure across a lot of the top universities, many of which who have actually joined into our third-line pancreatic program as well, but also the patient advocacy group said, "We believe in this type of a program and with what you're doing," and they're supporting our involvement in that trial financially to help speed that, hopefully to improve access to more patients and get new pancreatic cancer treatments on the market. So this has been really an important thing for us as a company, as well as getting access across all of the pancreatic cancer clinical trial sites.
Yeah, very important points.
Excellent, good. So, thank you. I'll wrap up just with touching on Ewing's just very briefly. We talked about the important partnership with Dr. Sant Chawla and the Joseph Ahmed Foundation. That study is ongoing right now. The study design is as follows. So there's 2 cohorts studying SM88 and 1 cohort, which is Ewing sarcoma. And Ben very nicely told the story around how we got to know Joseph Ahmed and his family. So that led to their interest in pursuing SM88 and Ewing sarcoma. And then the other cohort is 12 patients in other sarcomas. So this will be a phase 2 that will give us the opportunity with the primary endpoint of response rate to understand the efficacy and safety profile of SM88 in sarcoma. So just to conclude, so when you think about our 2020 focus, really falls into 3 key areas. Our clinical trial milestones are of utmost importance to us. And, to date, we've already achieved 2 very important milestones in terms of advancing enrollment in ADA PANC, and also advancing enrollment in our sarcoma study, which is referred to as HOPES. Another very important milestone, which was achieved earlier this year, was to initiate enrollment in PanCAN's Precision Promise study, which is a very prestigious study, as Dr. Del Priori covered. And then, as you can see, the rest of the year is really going to be focused on advancing our clinical program into other areas of unmet need, whether that's other solid tumors such as breast and prostate, and or potentially hematologic malignancies. And in terms of preclinical, we talked about, the importance of our preclinical data. We anticipate presenting our preclinical data for SM88 in the near future, in addition to Tyme 18, and also publishing some of the completed phase 2 work. So just in conclusion, we're very proud at Tyme of the advancements we've made so far, leading the scientific and clinical research in that next generation cancer metabolism-based therapies. Our lead therapy, oral SM88, we demonstrated encouraging efficacy across 15 different tumor types, both solid tumors and hematologic malignancies. We are passionate about advancing oral SM88 and our other therapies for patients with great unmet medical needs. So I thank you all for your participation today. And at this point, I will turn it back over to Mark.
Great. Thank you so much, Michelle and team. Really wanted to thank Ben, John, Giuseppe, Michelle for the incredible presentation. Always inspirational hearing it from you. Now we'll start with the Q&A session. We'd like to introduce Dr. Sean Fee. Give me just 1 second. I want to just share this screen if I may. If you have any questions, please raise your hand. We had some preliminary questions come in already. But with regards to introducing Dr. Sean Fee of MSQ, he has 15 plus years of industry experience, industry operating investments experience in the healthcare sector, including experience in private equity, venture capital, and mergers and acquisitions. He previously was a founding member of the Healthcare Fund, an adjunct professor at the National School of Development at Peking University. Dr. Sean Fee, please start with the Q&A and maybe introduce yourself. Thank you.
Yeah, thanks. Thanks, Ben, Dr. Del Priori, Jonathan, and Michelle. Thanks for your presentation. And hi to all of the audience. And actually, I'd like to start out with 2 questions. One is the science question, and second is a business question. And after that, we'll pick up some of the questions from our audience. And my first science question is if the SM88 has broad therapeutic efficacy and without unnecessary off-target toxicity, how about the efficacy? Can you talk more in detail? And as pancreatic cancer is a very tough problem, and this cancer is really very bad. Many big pharmas put lots of money on it and failed. So why do you choose this cancer as a priority in the pipelines? Because I saw the oral pancreatic cancer drug is you got a lot of progresses in the pipelines. So this is my science question.
Thank you, Sean. This is John. I'll start with the first part of that question. So as we mentioned with the activity, we've had anti-tumor activity in 15 different cancer types. And these are confirmed by blinded radiologists, tumor shrinkage. So, one of the questions we have gotten before is, safety, efficacy or bad safety comes with efficacy, usually with cancer therapies, and that is unfortunately true. That's one of the things that our founder was trying to really get around, is how can you drive the efficacy of a drug without, sacrificing the quality of life of the patient. If you think back to the mechanism about how cancer cells have this voracious appetite for certain, molecules such as amino acids, that is something that our drug really leverages well. And again, there is a lot of literature and even an FDA-approved product based on this biology of cancer's hunger for amino acids. This FDA-approved product is for cancer imaging, not for therapeutic aspects of it, of the treating. But we've twisted that to say, okay, well, we don't want to just image it. We want to treat the cancer. How can we disrupt that cancer and take advantage of this biology that is both going to be safe as well as efficacious? So again, we do have lots of options, but when it comes to why start with pancreatic cancer, what I'll do is I'll turn it over to Michelle, who does have some background. I'll steal some of her thunder so she can get right into discussion, but she was working at Celgene for over 12 years and was involved in the launch of gemcitabine and Abraxane in pancreatic cancer. And so she certainly is very familiar with the development and profile of this space. Michelle?
Excellent. Thank you, John. So I'll build on something that John said around the unique mechanism of SM88 and then talk about the data for SM88 and what led us to pursue pancreatic cancer. So when you think about the disease etiology of pancreatic cancer, the tumor itself is a small portion of the actual mass. The tumor is surrounded by a thick fibrotic stromal mass. One of the hypotheses for why SM88 is working in pancreatic cancer is the cancer cell is pulling in SM88. The cancer cell is viewing SM88 as something it needs. It needs non-essential amino acids. It's not realizing SM88 is a modified dysfunctional tyrosine. And also the fact that SM88 is a small molecule could be beneficial in terms of SM88 being able to reach the tumor. In terms of what led Tyme to pursue SM88 for pancreas cancer, the initial movement into the phase 2 was based on encouraging data from both the first in human and the compassionate use program. But we received very, very clear feedback from our world-renowned medical advisory board and guidance from the FDA that brought us from our phase 2 data into our pivotal stage. And the other very encouraging support for SM88 was an independent group with the steering committee for the Pancreatic Cancer Action Network choosing SM88 as the first experimental therapy for their very prestigious Precision Promise study. So all of those coupled together in terms of the mechanism of action, the data we saw, and the feedback from this world-renowned group of thought leaders led us to move forward into our pivotal program for pancreatic cancer.
That's great. And there's one other point I wanted to bring up, just because I noticed some of the questions coming in through the chat that relate back to what John was talking about with the mechanism, and also indirectly what Michelle was saying with pancreatic cancer. There's a fundamental aspect, and one of the questions was, why tyrosine, why not glutamine, why not a different amino acid? Another one was really asking about what's specific about the different cancer types. On a very basic level, we know all of the cancers have a much higher metabolic uptake of amino acids. So you're right, you're going to get more glutamine in there as well as more tyrosine. Now what's different is that tyrosine in a normal cell is used very sparingly. There's minimal uptake of tyrosine because most of the cells are making it from phenylalanine already. And so cancer actually has this different profile, and it's across cancers. That was what John was saying with the Warburg effect, something that was discovered 100 years ago, that they have this altered metabolism. They're actually going to take up many non-essential amino acids that normal cells will either not take up or take up sparingly. And so that's what gives us that specificity, is that we're able to take an amino acid that is either not or barely being used by normal cells, but is used in large quantities by cancer cells, and target those cancer cells specifically. And that's true across different cancer types. So I just wanted to, John, maybe you can talk about that a little bit more, since we were getting a couple of questions on the chat. I thought it was important to hit on.
Yeah, I mean, the whole glutamine question, for people who follow it, very, very astute. Glutamine probably is the #1 amino acid used by cancer cells. So it would think that it would be a very attractive target, and a lot of people have studied glutamine metabolism in cancer. The only problem is, or the problem that seems to have emerged from the work in that, is that unfortunately normal cells also use glutamine, so the off-target side effects have been limiting in how to effectively target glutamine to date. That's why, so tyrosine might not stick out as the most logical one. Glutamine would float to the surface, but that off-target toxicity to date has been the biggest problem with targeting glutamine. And so that's why using something and not focusing on what's the highest use, but what's selectively used by the cancer is how we approach this problem.
And Giuseppe, maybe you want to follow up the other chat question on the SAEs being related to study drug, because I think that sort of shows how selective SM88 is being taken up.
Yeah, thanks, Ben. So in two very different cohorts, in all 5 cohorts, we had no serious concern of a drug-related pattern of toxicities. But in the 4 cohorts that we've reported, and obviously we report them real time, we've seen no off-target effects typical of hormonal-based therapies. So in our prostate cohort, which is a very elderly group of people with multiple comorbidities and multiple medications, or in our pancreas cohort, which is extremely frail group of individuals. We see none of those off-target effects that you'll see from indiscriminate cytotoxic, or even when you see targeted therapies showing multiple organ, multiple tissue effects as well. So, based on radiolabel trials that we were alluding to earlier, we think that this is highly specific for metabolically active cancer cells and sparing normal cells, and that's reflected in our safety profile.
Thanks, Tim, for the very detailed explain. Actually, like you mentioned about the amino acid, the tyrosine. Actually, I have questions. There's about many amino acid. Like we have 20 very basic amino acid, and we also have the other amino acid. How do you choose the tyrosine? What I mean is that will you find the other amino acid in the future? And if you have a target amino acid, how do you modify it?
So, I mean, when it comes to, I think when we were focusing on which one to which amino acid to look at, the focus was what we were saying before, which one is the most selective to just the cancer. So you are right. There are other non-essential amino acids. Amino acids are taken in by the cells, and specifically cancer cells, by a variety of different channels and transporters. We found that tyrosine, based on the literature, and this is also what drove a lot of the imaging work, seemed to be the one that was the most selective. Like, again, in the imaging, it has the lowest background compared to some of the other amino acids that they tried to radiolabel. So is it possible to use a similar approach with other amino acids? It might be, but tyrosine seemed to be particularly useful. One of the other aspects about why tyrosine that made it really interesting is that tyrosine is also used in the production of certain neurotransmitters, such as dopamine. It's a starting material for the formation of dopamine. So there is channels on the blood-brain barrier that allow tyrosine to get into the blood-brain barrier. And for many cancers, that's actually a very important aspect of being able to treat brain mets. Not for every patient, but when a patient has a brain met, that's a very difficult prognosis to treat. And it's something that not only we have the ability to treat, but we've also seen activity in brain metastases in prior patients. Hopefully that addresses some of your question. The chemical modifications are not hard to do. You just have to figure out which are the best ones biologically to generate the highest uptake. And so the chemical modifications that we have had in the literature have shown that they increase the uptake of the tyrosine even more so than normal natural tyrosine, so that from a competitive inhibition aspect. But also what can happen when you have these chemical modifications, it can further interrupt some of the intracellular processes that are used to dealing with the unmodified, and that modification can make it less available to some of the enzymes. For example, cutting off a tyrosine off of a messenger transfer tRNA. It's used to transfer these amino acids to the protein making machinery. So it's really not hard to do it, but you just have to figure out what's the smartest changes to make. I'll turn it back to you. Sorry if I was long-winded.
Okay, thanks. Term is very urgent. Actually, I have the business question. Actually, my business question would like to listening your comments on the AI drug discovery sector. As you know, use the AI method or use the AI way, we can find some very effective target. Like I know several companies, startup companies, they use the AI technology to find some peptide candidates. So what's your comment on AI drug discovery?
I'll start.
Would you try to cooperate with some companies?
Well, when it comes to the AI platforms that are around today, I think that, it's possible that you could use it to help, further screen other agents. I would think that the best way to use, it's not something that we have used to date. And since we have a drug that's shown this efficacy, our focus is to drive it forward. I would think the best way to leverage AI for our platform would be to find complementary mechanisms, because we have a drug that seems to have broad efficacy and is safe, and it seems to have a very different mechanism than most drugs. Would use AI to say, what other drugs that are out there could this be the most complementary to? Because combinations is going to be a very important strategy for SM88, and it has a great profile for such things. So AI would probably be best to say, what other mechanisms ours are disrupting, plus drug B, where we could really get the best synergy. So I think that's probably the best way that we would be able to use AI going forward. Hopefully that addresses your question.
Okay, thanks. Actually, we have Karen, they do AI technology. Maybe we can talk it later. Thank you. And then I'd like to leave it to Mark. Mark, would you like to talk to Andy?
Sure. We have a couple minutes. We're getting a lot of questions. I've been sharing it with the team. So I'll allow Ben and team to answer one of the other questions of your choosing. If you can just regurgitate the question and then answer it, I think we can end after you answer that question.
I think one of the most interesting ones, they're all good questions, but if,
right, that's tough.
If we only have time for one. So going off of what John
we can do two, Ben. We can do two.
Okay.
Yeah.
Well, going off of what John was just saying, planning for a combination trial with IO. So there is actually, we typically talk about the protein synthesis component to the drug. There is something potentially also with the immune modulation that could be an interesting component to our drug, because we do have a racemic and we are getting an effect off of both sides, we believe. So that might be an interesting place to spend a couple of minutes, because that's obviously a very hot area in drug development.
Do you want me to hit on that really quick? Just very quickly, teaser, there's probably more information on the web, or we could get to people later. But we have seen a reduction in catecholamine production. One of the side effects you do see with SM88 is a mild fatigue when you start therapy. The L modified is known to inhibit catecholamine. Remember, the dopamine and catecholamine production, tyrosine is a core process of that. Catecholamines are critical to the intratumoral immune modulation, specifically TAMs, or tumor-associated macrophages, and the shift from M0 to M2, which are immunosuppressive. Those are important for several different kinds of cancers, including pancreatic, but also breast and others. And so by inhibiting catecholamine production, you can actually shift that away from the M2s towards the M1s, which are the cancer-fighting cells. So it's something that's still in the works, but it could be a very interesting combination that could open up the door for potential IO combinations. More to come later this year on some of that.
Yeah, that's something we're very excited about. And maybe, Michelle, do you want to close? There's a bunch of questions on different timing milestones. Do you want to just run through the milestones upcoming that people can look forward to?
Sure, absolutely. So let me start with the pivotal Tyme 88 PANC study. We anticipate full enrollment by the end of this calendar year. We're oftentimes asked if the unfortunate outbreak of COVID-19 will impact enrollment. To date, the Tyme 88 PANC is a US trial. To date, the clinical trials in the US that are focused on oncology interventional studies, especially for great unmet needs, are still enrolling. And in the case of 88 Pank, we are still actively enrolling our study in the US. So we will certainly keep an eye on any potential impact of COVID-19, but we still anticipate full enrollment by the end of 2020 with data to follow in 2021. The other question that was asked was around prostate. So our phase 2 prostate study, Dr. Del Priore did present the final data at ESMO in September, and we anticipate publication to also be this year. And then we also do anticipate a publication or presentation within this year for the phase 2 portion, the part 1 for 88 Pank. So those are some of the key exciting clinical milestones, and then preclinical milestones will be additional presentations for SM88 preclinical mechanism of action data and also Tyme 18.
Great. Well, thank you very much for that, Michelle, Ben, Giuseppe, John. Really exciting work that you're doing. Congratulations on all your milestones. It is a pleasure and honor to be working with you. Dr. Sean Shi, thank you very much for hosting with us from China. I want to thank the audience members as well. Again, please contact MSQ or Tyme directly with any questions. We have a lot of follow-ups to make, so we're looking forward to sharing those answers and sharing time with you in China. Thank you.
Thank you, everyone.
Thank you, everyone. Thank you, Mark.
Thank you.
Shi.
Have a great day, everyone in China. Have a good night. Ben and team.
Thank
Good night.
Bye-bye.
Okay.
Bye-bye.
Bye-bye.